The story of my PhD is a story of two patients with radically different symptoms and a little fruit fly that is trying to help me understand the basic biology behind those symptoms. Our first patient is Dino. He is illustrated in the picture with the hand. Dino recently suffered a semi-terrible accident. With his hand, he reached for a doorknob, trying to open a door that was stuck. But the doorknob was very rusty, and unfortunately, Dino cut his hand. Now, if you could take Dino's hand and take a little drop of blood, put it under a slide and observe it under the microscope, you would see something remarkable. Dino's hand is trying to heal. You would see an army of cells trying to contribute to this healing. You would see cells trying to close the wound. You would see other cells that are amplifying the signal for pain so that Dino can hopefully go to hospital. You would also see cells looking for bacteria around, trying to prevent them from entering into the bloodstream. Our other patient has a completely different problem. His name is Muhammad Ali, the famous boxer. Muhammad Ali suffered from a condition called Parkinson's disease. It is a disease in which particular brain regions slowly start to shrink, leading ultimately to problems with motor control. So patients often have trouble walking, talking, even eating and breathing. And unfortunately, there's no cure for this disease. What was recently found was that the same molecules and same cells you can find in Dino's hand, where they are trying to help, are also found in brains with Parkinson's disease. Nobody really knows what they do because there's no cut, there is no infection that we know of, so we don't know what they're up to, but we do know that they can be damaging the brain itself if there's too many of them and for a very long time. The point of my PhD is to try to understand which of these cells and which of these molecules are the most damaging to the brain and why. To this end, I use Drosophila melanogaster, the common fruit fly, because this is an organism just similar enough to a human so that we can mimic all of these molecules, but also we can really rapidly manipulate them. We can hyperactivate them or tone them down and try to see how the organism is responding. So far, in our preliminary results, we could see that if we predispose genetically the fly to have a flood of these molecules, the damage in the brain gets bigger. Hopefully, understanding how this damage happens in the fly will help us create better therapeutics for Parkinson's disease.